what we do for the Cell and Gene Live events is we take some questions in advance from registrants. And one of the more broad questions that was asked, but asked quite frequently, is just the newer tools for translational research that have been adopted by leaders in the field as it applies to whatever it is you're doing in the space. And so I know we talked about it slightly at the top of the hour, but I was hoping you can give us a little bit more breakdown into any tools that you see, especially from a cellular therapy perspective, that are being adopted today.
Sure. I mean, one obvious example is minimal residual disease. And there's many different ways to measure that, many different assays. But there seems to be a lot of interest, particularly from the KOLs in MRD assays. And the gestalt of that is that KOLs like to see that by day 28, day 56, that there is no residual disease. And that is a harbinger, predictor, if you will, of a good, long, deep remission of the cancer. The challenge, of course, is that is not the standard of care yet. The standard of care are PET scans. So the patient comes back for a PET scan at day 28 or day 56, or longitudinally thereafter. And so it's been a challenge to get MRD qualified to the point where KOLs use MRD in place of PET scans. And as you can appreciate, a PET scan is pretty expensive, and yet payers willingly pay for that. So the challenge becomes, how do the big MRD providers break into that market and displace the PET scans that the oncologists have grown up and know and use regularly? Because as Doug alluded to, in a perfect world, you could theoretically get rid of the PET scans and all of the associated infrastructure that comes with it and simply rely on a peripheral blood test. Just go to a local clinic, have your blood drawn, and get shipped off to a lab, and remotely you could be told, theoretically, whether or not your cell or gene therapy has worked and your cancer is in remission and it's deepening and that you're headed in the right direction. And I think that gets to some of the regulatory barriers, but also just in the way that medical schools train oncologists. They train them on PET scans. Why? Because that's standard of care. They don't train on minimal residual disease. Why? Because that's not standard of care. And that's the real intersection between real clinical practice in the world we live in and the clinical practice envisioned by KOLs.
Gotcha. Okay. Thank you. And Doug, before we move on to the audience Q&A, I wanted to give you an opportunity to provide feedback on that same question. Like, where are we with new translational tools being adopted by various leaders in the field?
Yeah, I put a lot of thought into this when you sent us the original questions, and I think probably the one that comes to mind that is the most significant is more and more people relying on multi-domain responder indexes. So this is not completely transitional because most of these multi-domain responder indexes are going to incorporate clinical endpoints, but often you'll have biomarkers within those as well. So, and for people who are not familiar, multi-domain responder index means that you have an endpoint that is comprised of multiple endpoints. And instead of relying on just a single set of data for your patients, you incorporate all of those different endpoints into sort of an overall picture of how the patient is doing. And the reason to do that is because when you have complex diseases, especially where there's a lot of heterogeneity in the patient population, any one of those characteristics that's within the index may not necessarily show progress for that particular patient. It could be that particular indicator was never really that bad in that patient. Their particular disease phenotype didn't show significant disease progression in that one area. Or it could be that they've advanced to the point where that particular indicator is not going to get better. But maybe other indicators would change in that patient, whereas in another patient, the set of indicators that show benefit from the drug are completely different. So I think that's a really important tool. It's very challenging, I think, and that you have to have a very strong scientific justification to go to, regulatory bodies to talk about that. But it opens up the possibility to get much better efficacy data for these rare diseases where you might run a trial that has 6, 8, 10 patients, where looking at a single indicator might give you absolutely no chance of powering the study to the point where you could see a real response there. But if you use the MDRI strategy, you can potentially get more of a picture that, yes, maybe all the patients aren't moving towards efficacy in every one of these domains, but all of the patients are making progress in some of the domains, and you can pull that out with the appropriate statistical model using that MDRI strategy. I think that's really exciting because I think it opens up the possibility that companies could actually get drugs approved for some of these ultra rare diseases that really can only benefit from cell or gene therapy.